Hello everyone, welcome to PG Pathshala. In this video, I'll be covering Module 8 of Cellular and Molecular Biophysics. This module is on cellular differentiation. I am Ashik Sethi and currently I work at the All India Institute of Medical Sciences, New Delhi. Let us see the objectives of this module. At the end of this module, you should be able to explain the term cellular differentiation, list various levels of cell potencies, understand clinical aspects of cell potency, explain induced pluripotency, explain transdifferentiation and dedifferentiation, and explain the importance of cancer stem cells. Let us start with a conversation that occurred between an evolution skeptic and Professor J.B.S. Haldane. Professor Haldane was a renowned biologist of 20th century and he has made significant contributions to population genetics and chemical synthesis of life. Professor Haldane, even given the billions of years that you say were available for evolution, I cannot simply believe it is possible to go from a single cell to a complicated human body with its trillions of cells that are organized into bones and muscles and nerves, a heart that pumps without ceasing for decades, and miles of blood vessels and kidney tubules, and a brain capable of thinking and talking and feeling. The reply to this question by Professor Haldane was too simple. He said, "But ma'am, you did it yourself and it took only 9 months for you." Why is it that a single cell should give rise to different cell types with diverse functions? What is the advantage of having multitude of cell types? What are the inadvertent consequences of a precursor cell giving rise to a phenotypically different progeny? All these questions will be addressed in this module on cell differentiation. The mechanisms behind cell differentiation will be discussed in a separate module. Life originated around 3.7 billion years ago in the form of single cellular organisms. Unicellularity is a successful trait and unicellular organisms still continue to exist, that too in abundant numbers. The first multicellular organisms evolved only around 600 million years before, that is much later than the unicellular organisms. Formation of multicellular organisms was a key moment in the course of evolution, and the key feature of multicellular organisms was cooperativity between various cell types that were phenotypically different. This prevented independent existence of individual cell types in such organisms. This also meant that a single cell type gave rise to multiple cell types that are distinct from the parent cell. Such a process in which a cell changes from one form to another is known as cell differentiation. Differentiation usually provides a cell with new functional capabilities. It is associated with extensive reprogramming at the transcription level, thus enabling expression of different sets of proteins at different levels in differentiated cell compared to the parent cell. So let's see an example of cell differentiation. You can see a hematopoietic stem cell giving rise to common myeloid progenitor and common lymphoid progenitor, ultimately leading to the formation of a multitude of cell types including basophils, neutrophils, eosinophils, monocytes, B lymphocytes, T lymphocytes, natural killer cells, and macrophages and dendritic cells, and even platelets. So if you see all these cells, they carry specific functions and they are different from each other. For example, platelets are involved in blood clotting, lymphocytes are involved in immune defense, and erythrocytes are involved in carrying oxygen to the peripheral tissues from the lungs. But it is to be noted that not all the cells in the body are capable of differentiation, and not all the cells that can differentiate do so at an equal level. This brings us to an important concept, cell potency. Cell potency refers to the ability of a cell to differentiate into different cell types. More the ability of a cell to differentiate into different cell types, greater is its cell potency. So based on potency, cells can be totipotent, pluripotent, multipotent, oligopotent, or unipotent. Let us see them one at a time. Totipotent cells are capable of generating all the differentiated cells in the organism. Zygote is a classical example of a totipotent cell. Zygote is formed after fertilization and it can generate all the cells that constitute the particular organism. Down the queue, we have pluripotent stem cells. Pluripotent cells are capable of generating cells of all the three germ layers, that is ectoderm, endoderm and mesoderm. An example of pluripotent stem cell is the embryonic stem cell that is present in the inner cell mass of the blastocyst. The blastocyst is usually formed after 5 to 6 days of fertilization. What differentiates totipotent cells from pluripotent cells? Totipotent cells can give rise to the whole organism and also extra embryonic tissues like placenta, whereas pluripotent cells cannot form extra embryonic tissues. Since pluripotent cells cannot form placenta or extra embryonic tissues, these cannot develop into a fetus, whereas totipotent cells can form a fetus. So that is the difference between totipotent and pluripotent cells. Let's now see multipotent cells. Multipotent cells can give rise to certain discrete differentiated cell types but are restricted in their cell potency as they cannot differentiate into all cell types. Examples of multipotent cells include mesenchymal stem cells that can differentiate into various tissues like adipocytes and fibroblasts, and also hematopoietic stem cells that we have just seen, which can differentiate into lymphoid and myeloid lineage and to a multitude of cell types. Oligopotent cells have a further restricted potency and can generate a few cell types compared to multipotent cells. We just saw lymphoid and myeloid progenitors, so they are examples of oligopotent cells. Unipotent cells can give rise to only one cell type. What is the difference between a unipotent stem cell and tissue cells? Unipotent cells can self-renew. This is not a property that is shown by tissue cells. This image summarizes cell potency. A totipotent stem cell gives rise to pluripotent embryonic stem cells that are capable of differentiating into all the three germ layers, that is endoderm, mesoderm and ectoderm, which give rise to multipotent stem cells that are restricted in their potency compared to pluripotent stem cells. And finally, you have your adult cell types that are generated, giving rise to different cells that are present in different organs. Let us now see some of the clinical aspects of cell potency. Hematopoietic stem cell transplantation is a treatment modality available for various disorders like leukemias, blood disorders and certain genetic disorders. Let's see the procedure of hematopoietic stem cell transplantation with the help of an image. You have a donor organism and a recipient organism. The recipient organism is irradiated so as to destroy all the blood cell progenitors that are present in the recipient organism. Hematopoietic stem cells, that are the multipotent cells, are harvested from the bone marrow or the peripheral blood of the donor. Now these are introduced into the recipient. Once the transplantation is complete, these hematopoietic stem cells will regenerate the hematopoietic cells in the bone marrow of the recipient. Let us see a therapeutic application of hematopoietic stem cell transplant. In lysosomal storage disorders, hydrolytic enzymes that are involved in the breakdown of complex molecules are defective, and this leads to dysfunction of lysosomes and eventually other organelles, leading to cytotoxicity. The brunt of the attack is borne by macrophages, which are the scavenging cells. Treatment for such disorders, till the advent of enzyme replacement therapy, included transplantation of hematopoietic cells, which ultimately differentiate into various lineages, including macrophages, so as to restore the normal cellular function in the recipient. A similar approach has been considered for gene therapy of single gene disorders. Here, the defective gene is corrected or replaced in the hematopoietic stem cell, which is later reintroduced into the affected individual. This should enable a lot of cells to express the normal protein and thus cure the genetic disease. Another clinical application is teratomas. Teratomas are tumors that have components of more than one germ layer. These usually arise from pluripotent cells of testis or ovary or aberrantly migrated cells. Such malignant cells have the potential to differentiate into multiple adult tissue types and can give rise to tissues like bone, hair, and teeth. Histological grading of tumors. This is based on the degree of differentiation observed in the tumor tissues microscopically. Tumors are usually classified into well, moderate, or poorly differentiated, or grade 1, 2, 3, or 4, or low or high grade. Anaplasia refers to the lack of differentiation of tumor cells. Increase in anaplasia is associated with a high aggressive tumor and a bad prognosis. Tissues are classified based on their proliferative capacity into labile tissues, stable tissues, and permanent tissues. Labile tissues are continuously replenished by a pool of tissue stem cells. Examples include blood cells, intestinal epithelial cells, and skin cells. Such tissues have a high regenerative capacity after tissue injury. On the other hand, stable tissues have a minimal capacity of proliferation and a minimal capacity to regenerate following injury. Most of the solid organ parenchyma is composed of stable tissues. Permanent tissues have almost no proliferative capacity. Example, neurons and cardiomyocytes. Thus, injury to these tissues cannot be completely recovered if the injury results in cell death. This emphasizes the concept of golden hour in diseases like stroke and myocardial infarction, where an immediate medical intervention can minimize tissue injury and prevent irreparable damage. So those are all the clinical aspects related to cell potency. Let's now discuss induced pluripotency. Development of an organism is associated with reversible changes in the epigenome. Studies have shown that it is possible to reprogram an adult tissue cell to a pluripotent cell by inducing the expression of various transcription factors. At the turn of the millennium, Yamanaka and Takahashi identified 24 transcription factors that are related to pluripotency, and they screened these factors to eliminate redundant factors. They finally identified 4 genes that alone can induce pluripotency, the genes being OCT4, SOX2, KLF4, and c-MYC. These are called Yamanaka factors. The induced pluripotent stem cells thus formed could not be reprogrammed fully to an embryonic stem cell phenotype, and these were called the first generation induced pluripotent stem cells. The first generation induced pluripotent stem cells used a specific embryonic stem cell protein as a marker, and that was FBX15. In second generation induced pluripotent stem cells, the same transcription factors were used, but the selection marker was changed to a different protein called NANOG, which is another embryonic stem cell specific protein. These cells almost mimicked embryonic stem cell. Yamanaka was awarded Nobel Prize in Physiology or Medicine in the year 2012 for his discovery of induced pluripotent stem cells. There were multiple ethical issues with regard to the usage of embryonic stem cells in research, as certain religious and scientific friends presume that isolation of embryonic stem cells from human blastocysts involves destruction of life. Induced pluripotent stem cells are considered to be an alternative to this. Another potential use of induced pluripotent stem cells are in autologous transplantations, where somatic cells from an individual can be used to generate induced pluripotent stem cells and can be differentiated into different tissue lineages based on the need. A major caveat of induced pluripotency is that the transcription factors that are used in inducing pluripotency are products of oncogenes, and overexpression of their gene products might drive the cells into abnormal proliferation and development of malignancy. Despite such challenges, induced pluripotent stem cell technology can have multiple clinical applications, and this has been exemplified by the use of induced pluripotent stem cells in generating retinal pigment epithelial cells to treat retinal degeneration. This study was conducted in Japan in the year 2017. During dedifferentiation, a terminally differentiated cell reverts back to a cell primitive in the line of differentiation, and this primitive cell has the potential to proliferate and give rise to the cell type these arose from. This is exemplified by Schwann cells present along the neurons. Mature Schwann cells do not proliferate, but once they are detached from the adjoining axon, they de-differentiate and express genes that enable proliferation. In transdifferentiation, cells switch lineages to form a different cell type. This can be induced using various approaches and can lead to generation of closely related cells from a different lineage. Example, macrophages and B cells. What are the other implications of de-differentiation? Let's talk about cancer stem cells. Cancer stem cells or tumor initiating cells are the cells with stem cell-like properties, that is self-renewal and ability to differentiate into tumor cells. The bulk of the tumor tissue is formed by terminally differentiated tumor cells, and cancer stem cells usually comprise only of about 1% of the total tumor population. However, due to their proliferation and differentiation capabilities, they can lead to generation of new tumor cells. So in this picture, you can see a cancer stem cell that is depicted in red color. The cancer stem cells leads to production of two types of cells. One is the tumor cells and other one is by self-renewal, it can regenerate itself to form cancer stem cells again. That's again depicted in red. And if you see the entire tumor bulk, the population that is in red is very minimal compared to the tumor cells that are present. That's why it said that cancer stem cells, they form only minimal percentage of the total tumor bulk. The bulk of the tumor tissue is formed by terminally differentiated tumor cells, while cancer stem cells comprise of only about 1% of the total tumor cells. However, because of their proliferation and differentiation capabilities, they can lead to generation of new tumor cells. Two major hypotheses has been put forth to explain the development of cancer stem cells. They could arise from mutations building up in tissue stem cells, which are oligopotent as we have seen earlier, thus retaining their self-renewal and differentiation capabilities, but driving differentiation along an aberrant pathway leading to malignancy. The alternative hypothesis suggests that cancer stem cells arise due to de-differentiation of mature tissue cells, enabling them new capacities of proliferation and transdifferentiation. Conventional chemotherapy regimens used in the treatment of cancers target tumor cells and can help in reducing the bulk of the tumor. However, cancer stem cells are resistant to chemotherapy, and until unless they are eliminated, new tumor cells will continue to form. Thus, cancer stem cells are considered to be responsible for tumor relapse after treatment and also for metastasis. Let's see an example. Chronic myeloid leukemia, CML, was the first malignancy attributed due to a chromosomal abnormality as a molecular defect. You all should be familiar about Philadelphia chromosome. Philadelphia chromosome leads to the formation of BCR-ABL fusion protein, which is an abnormal tyrosine kinase. So CML was the first malignancy in which an abnormal tyrosine kinase, that is a BCR-ABL fusion protein, was identified, and indeed the first malignancy in which a tyrosine kinase inhibitor was found to be effective. You all should be familiar with imatinib, which is a tyrosine kinase inhibitor. However, patients with CML require a lifelong therapy with imatinib. Any attempt to stop the treatment with imatinib will result in relapse of the condition. I hope you will be able to answer why there is relapse of the disease on stoppage of imatinib treatment. Those who could figure out the answer, congrats. The answer is imatinib only targets the tumor cells. Cancer stem cells are spared. So on stopping the treatment, new tumor cells arise from cancer stem cells and thus lead to relapse of the tumor. To summarize this topic, the process in which a cell changes from one form to another is known as cell differentiation. The ability of a cell to differentiate into different cell types is known as cell potency. Cells can be totipotent, pluripotent, multipotent, oligopotent, or unipotent. SOX2, OCT4, c-MYC and KLF4 are known as Yamanaka factors, and these are involved in induced pluripotency. Transdifferentiation and dedifferentiation have a potential application in regenerative medicine. Cancer stem cells can derive from abnormal adult tissue cells and can lead to tumor propagation and chemoresistance. That's all for this module. The mechanism for cell differentiation merits a separate discussion in detail, and that will be covered in a different module. Thank you.